All right, let's dive into the incredible world of bioinformatics. This is where biology meets computer science, where we use massive amounts of data to solve some of the deepest mysteries of life itself. So it all kicks off with something that looks a little like this, just a huge, seemingly random string of 4 letters, over and over again. This right here is the raw code of life, DNA. But on its own, it's just data, right? It's a total mystery. It's like someone handed you a book written in a language you've never, ever seen before. So that leads us to the big question. How do we get from this jumble of letters to actually understanding diseases, or creating new medicines, or unlocking the secrets of how we evolved? How do we translate the book of life? Well, the way we do it is we become digital detectives. We're going to use some seriously powerful computational tools to follow the clues hidden inside this code, build our case, and hopefully solve the mystery of what this specific piece of DNA actually does. So let's get our investigation started. Okay, first things first. As any good detective knows, your first job is to identify the suspect. We've got this unknown sequence, this string of genetic code, and we need to put a name to it. This is step 1. We've got to crack the code. And our number one tool for this investigation is a program called BLAST. Now, the easiest way to think about BLAST is like a search engine. But instead of typing in words, we paste in our mystery gene sequence. It's basically Google, but for genomics. It's that powerful. So what BLAST does is it takes our query sequence, that's our unknown on the left, and it just scours these massive global databases, comparing it against billions of known sequences from all kinds of organisms. It's looking for a match, a genetic fingerprint that's already on file. And look at that, we got a hit. Our sequence is a perfect match for something called human gene XYZ. But hold on a second. We're talking about a database with billions and billions of letters. How do we really know this match isn't just some random fluke? I mean, how can we be sure this is a genuine lead and not just a dead end? Well, this is where a little bit of statistics comes into play. We use this statistical measure called an e-value, which stands for expect value. Honestly, just think of it as a coincidence score. It tells us how many times we would expect to find a match this good just by pure chance. So a super, super low e-value, something close to 0, tells us there's no way this is a coincidence. Our match is legit, it's highly significant, the investigation now has a solid ID. So we've identified our gene, we've got a name for our suspect, that's huge, but that's really just the blueprint. Now we've got to figure out what kind of machine this blueprint actually builds. And more importantly, what does that machine do inside the cell? It's time to uncover its function. And to do this, bioinformatic detectives use this really clever principle called guilt by association. It's just like in a classic crime drama, right? You can learn a whole lot about a suspect by looking at the company they keep. If our mystery protein is always hanging out with a known crew of troublemakers, well, it's probably involved in the same kind of trouble. One of the absolute coolest methods for this is called the Rosetta Stone approach. Let me walk you through it. In one organism, you might find protein A and protein B floating around as 2 separate things. But then, in a totally different organism, scientists might find them actually fused together into one single combined protein. The insight here is brilliant. Evolution wouldn't bother sticking them together unless they work together. This is a massive, massive clue that A and B are part of the same team. Another really powerful technique is called phylogenetic profiling. Now, this is where the detective work kind of goes international. We're tracking our protein suspects across the entire tree of life. We look at a whole bunch of different species, say E. coli, a worm, a plant, and we just check to see if they have a version of our proteins. And if you look at this table here, you'll see something pretty interesting. Proteins P2 and P3 have the exact same pattern. They either show up together or they're both gone. And that is not a coincidence. What it tells us is something really fundamental. Proteins that work together evolve together. It's like a package deal. If you need P2 to do a job, you're also going to need P3. If you lose one, you might as well lose the other. This coevolution pattern is another really strong piece of evidence that they're on the same team. So, you see what we're doing here? We're starting to build a network of associates, a social circle for our protein, because no protein works alone. It's not an island. It operates as part of this incredibly complex social network inside the cell. And by mapping all these connections, we can start to see the bigger picture of its role. So let's just quickly recap our detective's toolkit. We started by getting an ID for our sequence using homology with a tool like BLAST.
Then, we started finding its functional partners, its crew, using the clever Rosetta Stone method and by tracking its evolutionary footprint with phylogenetic profiling. Each one of these computational methods gives us another line of evidence, helping us build a really strong case for our gene's function.
Okay, now for the payoff. How does all this digital detective work, all these clues and networks and associations, how does this actually solve a case in the real world? This is where we finally go from just data to actual diagnostics.
By combining all these different layers of data, scientists can build these unbelievably detailed profiles. They can literally see which genes are turned on or off in a diseased cell compared to a healthy one. And this lets them link entire networks of genes, not just one single suspect, to really complex diseases like Parkinson's or Alzheimer's. And that opens the door for brand new computational diagnostics and much more targeted treatments. Ultimately, this all comes back to the core principle of good detective work. As this quote says, real confidence comes from the convergence of multiple analyses. It's never about just a single clue. It's about building a solid case where multiple independent lines of computational evidence all point to the very same conclusion. So, through the power of bioinformatics, the book of life isn't written in some alien language anymore. We can actually read it. It's becoming more and more clear every day. The real question now is, with this incredible power to ask almost anything of our own genetic code, what should we be asking it next?